Scott, thanks very much. And Dr. Fauci, thank you so much for joining us today. You know, everybody wants to hear your opinion on the data from the Moderna coronavirus vaccine trial, the early data we saw this week. You said you're cautiously optimistic. Tell us about your reflections on that data.
Well, as you know, when you do a vaccine trial, there are various landmarks along the trial that could actually derail it at any given time. The first that we saw was we went into a phase 1 trial very quickly, really with record speed. And we did it with 3 doses. And the first thing you look at is it's safe. Clearly it was. But the important issue is it inducing the kind of response that you would predict would be protective. So when the company and our investigators looked at the data in the first 2 doses of the phase 1 study in normal volunteers, we found something that was really quite encouraging, in that the immunizations induced not only antibody in general, but neutralizing antibody. And for people to understand what that means, if this were the virus here, it means if this were the virus here, and you had an antibody that bound to one part, it would bind like maybe to this part here, which is not neutralizing because the business end of the virus is here. So the antibodies that were induced were ones that actually block the component of the virus that binds to your cells. That's really good news, because not only is it the right antibody, but the titer of the antibody that was induced was really very good with the standpoint of what you predict might be protective. So even though it's a small number of individuals and it's the first step in a multi-step process, it was still very encouraging. We still have a long way to go, obviously. There are so many things that need to be done. We're going to go quickly into a phase 3 trial, probably in the beginning of the summer, sometime in July. And then the proof of the pudding will be, is it effective? Namely, does it really protect against infection? But the first stage has really been encouraging.
And the data you're talking about, the neutralizing antibodies, there were only 8 people's worth of data available on neutralizing antibodies as of this cut of the data. And all 8 did show those antibodies. Can you tell us any more about those people? You know, some folks pointed out if they were young, relatively, healthy, robust people, that might be different than if they were older folks.
Yeah, I mean, the phase 1 trial has certain constraints in it that you have to get it if you really want to do a phase 1 trial properly with young, relatively, more than relatively, but to all appearances, quite healthy individuals. So they were individuals from the age of 18 to 55. I can't tell you what the age was of the individual people there, but it was within that range of 18 to 55 years old. As we go further in the study, we'll be doing older individuals, and then we'll be doing children, and then we'll be doing other subpopulations. But the first shot at the phase 1 were individuals who were young and otherwise healthy.
And this data from Moderna wasn't the only vaccine data we've seen in the last week. We've seen some animal data from the Oxford-AstraZeneca trial. We just saw data published in The Lancet from CanSino Biologics out of China from a human phase 1 trial. All of that combined, how has your confidence level changed this week, if at all, that we will have a successful vaccine for COVID-19?
Well, whenever you're dealing with vaccines, you've always really got to be conservative about it, because the history of vaccinology is one that is punctuated by as many failures as that there are successes. But we have what I call multiple shots on goal. It isn't just one candidate. The one I mentioned was Moderna, but by no means are they alone. There's the Oxford chimp ad that we spoke about, that you just mentioned. And then there are a number of other companies that are in various stages of development, and they will be going into phase 1 trial in a staggered way sometime as we get into the mid and late summer. That's a good thing. The United States government, my institute that I direct, is going to be involved one way or another, directly or indirectly, facilitating many of those trials, either by getting and allowing our clinical trial sites and the apparatus that we set up over many years to make it available to these companies. So I'm always cautiously optimistic when I see a number of companies that are really quite good. They have a good track record. You have the backing of the federal government, and the early results look encouraging. Again, I don't want to jump ahead of myself, but those are the kind of things that make me feel cautiously optimistic that within that time frame that I've spoken, that we will have a safe and effective vaccine with no guarantees, because again, you never know if it's going to work or not, but we have some cautious optimism.
And these timelines, of course, are faster than pretty much anything we've ever seen in history. So that being said, what threshold are you looking for in terms of how much information is needed on efficacy and on safety for, you're not the regulator, but the regulators will require to approve these vaccines?
Well, I mean, the benchmarks and the things that you're looking for, obviously, is you want an efficacy signal. I mean, the studies that have been designed are randomized placebo-controlled trials in which you look at the vaccinated versus unvaccinated and you see if you get an efficacy signal. We're going to be doing a lot of people, and in each arm of a trial, you can have, tens of thousands of people, anywhere from 10, 15, 20, 30,000 people per arm of a trial. And in fact, if you do that, hopefully that you'll get enough information quickly enough that you'll be able to make a determination not only of the safety, but of the efficacy of this. And the sooner we do it, the better. One of the things that I do want to bring up is the reason we're able to do this more quickly than we've done in other situations without compromising safety and without compromising the scientific integrity of the study is that we're doing things at risk. In other words, rather than the standard way of you go from step one and you don't make an investment for the next until you're sure of the step one, then you get to step two, you don't prepare sites, you don't start making enough doses until you know it works. When you proceed at risk, you make the financial investment assuming that you're going to be successful. So when we say at risk, we don't mean at risk to the patient. We mean at risk to the financial investment, so that it could be that if you're not successful, you made major investment in resources that you may not be able to recover. Whereas if you are successful, those investments that you made in the resources to produce and to prepare sites actually will put you months ahead of where you normally would be. So I want to make sure that the public understands when we talk about that, we're not compromising any safety, nor are we compromising the scientific integrity of the study.